Alright, hey AP Bio 2, this is going to be the second video in Unit 3, Cell Structure and Function. So we're going to pick up where we left off, which I believe was talking about bacteria. We're going to start on this slide, slide number 6. So get your notes ready. Here we go. Alright, we've talked a little bit about prokaryotes already this year, so now we're going to apply that to comparing and contrasting them to eukaryotic cells. So first off, typically prokaryotes are smaller than eukaryotes. I mentioned in the last PowerPoint that they are between 1 and 5 micrometers versus a eukaryotic cell being 10 to 100 micrometers, which is quite a bit bigger. Even though all cells are small, please don't think that it's that big. I'm just saying, relatively speaking, if you're talking about micrometers, bacteria are much smaller, and we also mentioned how that means that they are much more efficient at getting materials into the cell and out of the cell, and also like within the cell, moving them around. Okay, don't forget bacteria belong to two kingdoms, kingdom Archaebacteria, kingdom Eubacteria. Remember, Archaebacteria are the ancient ones found in harsh conditions. Eubacteria are the ones that are more common today to you and me. This is where some of the pathogenic examples go, but remember that they're not all pathogenic because we have some great decomposers in that kingdom as well. A little bit of extra info right here. Remember, bacteria, all prokaryotes, have no true nucleus. In fact, the word pro means before. And if you put the word before by the word karyon, which means kernel, which is like a colonel in an army, that means before a kernel was present, that kernel is that control center. Okay, control center. So before that control center was present, that's what a prokaryote is. No true nucleus, no membrane-bound organelles. Okay, so this was something that my freshmen struggled with. When I say no membrane-bound organelles, that means no organelles that are bound by a double phospholipid bilayer, because that's what membrane-bound means. It's got a double phospholipid bilayer that separates that organelle from everything else. So no true nucleus, no membrane-bound organelles. They do have DNA, and we will refer to that as chromatin quite a bit, which is DNA plus protein. Okay, that's what chromatin is. They do have that. It's not found in a nucleus. It's found in a nucleoid region, is what we call it. It's basically inside, you can see right in here where my arrow is, it's inside the cytosol, and we call that a nucleoid region where the DNA is. It's not floating aimlessly. It is floating in the cytosol, but it is in an area known as the nucleoid region. So no true nucleus, no membrane-bound organelles, yes on the DNA. They are also going to have ribosomes, which are the dots that are used for making proteins, and we'll talk a little bit more about some things that they have on the next slide. Okay, so here's the structure of a typical prokaryote. Okay, a typical prokaryote is going to have cell wall. All bacteria have a cell wall. In fact, you already know, if it was in kingdom Eubacteria, what is that cell wall made up of? That's just a review, right? Peptidoglycan. Hopefully you can remember that. Peptidoglycan. If it was in Archaebacteria, it was polysaccharide material. Okay, it's a polysac. So we know all bacteria have a cell wall. They do not all have a capsule. This is for some, a capsule. That's for some, and we already talked about in Unit 1 how that capsule, okay, let me put some, that capsule can be slimy, and that can also help them gain entrance into the host cell, but that also it protects them from, like, let's say in our bodies, from our immune system being able to tear them down. All bacteria have a cell membrane. In fact, all cells have a cell membrane because the job is to regulate homeostasis by letting some things go in and some things go out. So that's to all cells. Yes, they have chromatin. That's DNA plus protein. Okay, and all cells have that as well. In fact, that's not only unique to all cells. Remember, viruses, which are non-living, also have DNA material. Okay, ribosomes are these little dots that you can see in here. Okay, it's number 5. That's the ribosome. Pili are these little things sticking out that they use in sexual reproduction. Okay, that's for their sexual reproduction. Flagella are used for movement. Okay, movement. So you can see that the typical prokaryote is very small. but they're still going to have some structures to them, and I'm going to add one on the list. I'm going to point to this space right in here, if my pen will allow me to. Let's try this right in here. We're going to also add the word cytosol. Cytosol, which is cell gel. Okay, we'll talk about that when we get to the eukaryotic structures as well, but I did want to point that out because that's the jelly-like fluid that is inside of the cell that suspends the ribosomes in the case of the bacteria. Okay, so cell wall might be made of peptidoglycan, might be polysaccharide depending on the kingdom. Capsule is in some bacteria, so it will be on the outside of that. You can see in this picture it is the yellow. Cell membrane would be the innermost membrane, regulates homeostasis, found in all cells. Chromatin is DNA protein, that's this purple stuff in the middle. Ribosomes are the dots that are used in making proteins, and even though they are bacteria, they must have proteins. So that's to make protein, I'm going to add that there. Pili are for sexual reproduction, they're all the hairy-like structures on the outside, and then flagella is for movement. Okay, that's your typical prokaryote. Now, for us, and not just for us, for plants, for protists, for fungi. So now the rest of this unit, we're going to be talking about eukaryotic cells and all the organelles that are in a eukaryotic cell. So they are larger, okay, more complex than a prokaryote. They do have a true nucleus. If you look at the word "eu," this is one of your root words. EU means true. And then karyon refers to the kernel, so this is a true nucleus. What that true part means, I'm going to add up here, means bound by a nuclear envelope. Okay, because true nucleus means has a membrane-bound nucleus. The membrane around the nucleus is known as the nuclear envelope. Okay, not only does the eukaryotic cell have a true nucleus, you're going to find a lot of membrane-bound organelles. Okay, remember an organelle is a little organ that has a certain function inside the cytoplasm. An example, you've heard of Mr. Mighty Mitochondria. You've heard of probably the ones down here as well, ER, Golgi, lysosomes, vacuoles. All of those little organelles are suspended in the cytosol. And the cytosol, let's just go ahead and define these two words really quickly, if that's okay. I'm going to define cytoplasm versus cytosol. And I did this freshman year, but I know it's been a long time, so I want to make sure that you guys know. Cytosol is the cell gel. That is literally, like if I draw a cell, let me draw my circle. Okay, if I point to that space, that's the cytosol. If I use cytoplasm, it's cytosol plus organelles. Okay? So cytosol plus the organelles make up cytoplasm. So cytoplasm would include the gel, plus it would include the ribosomes, plus it would include the mitochondria, plus it would include the Golgi apparatus. So everything inside of the cell, we would call that cytoplasm, whereas just the space is cytosol. So I'm going to be using those two words a lot. I just wanted to make sure I stopped right now and went over that with you. So if you look in this little bullet, it says eukaryotic cells have a true nucleus. They have membrane-bound organelles that are suspended in the cytosol. Okay, now if you look at the next little bullet, we're going to go into a tour of all of these. But in the organelles, we're going to talk about the nucleus. So we'll look at just that control center and go into that. Then we're going to focus on the ribosomes because those are also found in prokaryotic organisms. Then we'll focus on what is called an endomembrane system that's composed of the organelles that you see here, endoplasmic reticulum, that's smooth and rough, Golgi apparatus, lysosomes, and vacuoles. Those are all part of the endomembrane system. Okay, then we're going to focus on mitochondria and chloroplast, which are energy. Okay, these are energy transforming organelles. Okay, so we'll talk a lot about those. We'll get into the peroxisomes, and then we'll talk about the cytoskeleton, which is most of your cell structure and support. Okay, so that's kind of our tour that we're going to take in the next few slides. Before we do that, I thought I would show a picture really quickly so that you can see the structures are basically what you saw 9th grade year. Yes, you need to know what these structures are. Okay, so spend a lot of time going over these, but just to kind of show you, here's the nucleus in the center right here. And inside the nucleus, we have nucleolus, which is that dark, dense region that makes ribosomes. That's where ribosomes are born. Okay, then we have the squiggly lines in here is the chromatin. Okay, we've got the envelope that I'm outlining right here. That's the nuclear envelope around the outside. We've got nuclear pores right here that allow for the ribosomes and RNA to come out. Okay, so that's like all in the nucleus. Then we've got this folded back and forth right in here. This is rough, ER. You can tell it's rough because it has dots all over it, whereas this over here is smooth ER because it looks like this rough ER, but there are no dots on it. Okay, we've got Golgi apparatus right here. You can tell it looks like a flattened stack of pancakes. Okay, we've got mitochondria right here. This is an animal cell, so we've got centrioles right there, which are used in cell division. Okay, we're going to have, this one happens to have microtubules and microfilaments, which are part of the cytoskeleton, has microfilaments, microtubules, and intermediate filaments, which are the purple-looking structures that look like little spaghetti straws. Okay, we've got lysosomes. You can see it through here. There's another lysosome right over here. Okay, we've got ribosomes sprinkled throughout, all little dots. And what did I leave out? Oh, the centrosome it says that centrioles, that's what I call that. And this has a flagella on it. All human cells do not have a flagella. And then we're going to also talk about these little peroxisomes. Okay, so that's your typical animal cell. Look at that compared to plant cell. Okay, obviously we have another outer boundary on this, which is the cell wall. That gives it its more rectangular shape. Okay, you're going to also see chloroplasts right here. Okay, there's your chloroplast. Notice you do still see mitochondria. Plant cells have mitochondria and chloroplasts, which are both transforming organelles. We see a really big central vacuole. I forgot to show you, but an animal cell has vacuoles too, but it's really big in a plant. We still see Golgi. We still see rough ER, smooth ER. Okay, cell membrane right in there, cell wall on the outside, nucleus, nucleolus, chromatin, nuclear envelope. We still have the same things. Peroxisomes are available here. Okay, so we can kind of see, compare and contrast the two. Notice you do not see centrioles. Okay, centrioles are not present. And you do not see lysosomes. Okay, those are the two that you don't see in this picture. All right, here goes our tour. Okay, so we're going to start with the nucleus now and go through a little tour of the structures of the nucleus. This should mostly be review for you, so you should feel pretty confident as I'm going through this. But the nucleus is known as the control center of the eukaryotic cell. Why? Because it's the area, kind of like the air traffic control center tower at the airport, where it's going to direct all the airplanes. This is pretty much directing everything about your cell. It's all of your genetic information, plus it's driving how you produce your proteins, which expresses all of your genes. So it is known as the control center. So it's the whole thing that you see here, not the blue though right here. This is the rough endoplasmic reticulum, but this whole part right here, that kind of looks like a peach that was cut out, like a fruit, a peach, that's the nucleus. Okay, you can see the nuclear envelope is a double membrane. It is a phospholipid bilayer, but it's right here. It's got two membranes, one inside of the other one. Okay, you can kind of see in this picture right here, down here at the bottom. It's a double membrane that protects the nucleus, and it is continuous with the endoplasmic reticulum. If you notice, there's a little branching out like this, and then it's going to create the rough ER that branches off of the nucleus. There are pores in the nuclear envelope. You can see one here. I'm coloring it in blue. One here, one here. These are the little pores. Those pores, they're called nuclear pores, are going to allow RNA plus ribosomes to leave the nucleus. Now, in terms of why would RNA leave the nucleus, when we talk about protein synthesis, we're definitely going to need that to exit the nucleus to function in the cytosol. And then ribosomes, of course, to do their job, they're going to have to come out of the nucleus. And so a ribosome is made here in the nucleolus. You can see that's the next job right here. Put a star by that. The nucleolus is the site of ribosome production. So a ribosome is born here. It comes out of the nuclear pore like this. And then once it comes out, it can either attach to the rough ER, or maybe it comes out and floats in the cytosol, because ribosomes can be two places. They can either be free-floating or they can be bound to the rough ER, and we'll talk about that more in just a second, but I wanted to make sure you knew that the nucleolus is where the ribosomes are made. Okay, the chromatin are the squiggly lines that you see in here, and that, once again, is DNA wrapped in protein. Okay, so that's the nuclear structures. Okay, the ribosomes, we'll talk a little bit more about those here. Like I said a minute ago, this is a great picture, by the way. Look how cool this is, where you can see the pores. If the ribosomes come out, okay, of the pore, and they're free-floating, okay, we call those free ribosomes. If my pen will work, here we go. They're called free. And when they make a protein, the protein is going to stay within the cell. Okay, stay within the cell. So if it's a free-floating ribosome, comes out to the nuclear pore, floats out here in the cytosol, it makes a protein, but that protein's job is to stay within the cell. Okay, if it's a ribosome that comes out and attaches to the rough ER, we call that a bound, B-O-U-N-D. Okay, B-O-U-N-D. It's a bound ribosome, and that protein that it produces will be exported out of the cell. Okay, protein is exported. So there is a difference in the functioning of the ribosomes as to whether they are bound or whether they're free-floating. And here's a big picture of a ribosome. When we get into the chapter on protein synthesis, you will learn the name of the top and bottom. It's really cool, but there's actually two subunits. There's a top and a bottom that function together, so when they work in protein synthesis, like messenger RNA can be right here between the two, and the job can be done with that. So there's a large ribosomal subunit and a small ribosomal subunit that will do their job. So you do need to know the difference between a bound and a free ribosome. It has to do with the production of the protein, whether it's exported or whether it stays in the cell. Okay? All right, now we're going to get into the endomembrane system. The endomembrane system is including the structures that you see on this page. Okay, so let's just kind of look for a minute. Endomembrane, if you just break that word apart, use your root words. Endo means inside. Okay, so this is inside. And inside the membrane is the cytoplasm. So these are structures that are inside the cytoplasm that includes, okay, look at what it includes. It actually is going to include, I'm going to write up here, you would include the nuclear envelope in that, even though I talked about it on the slide before. We would include the nuclear envelope, or then including the rough ER, okay, rough ER. We've got Golgi apparatus. We're going to include that as well, Golgi. We're going to include the smooth ER, because rough and smooth are both endoplasmic reticulum. We'll include the lysosomes, S-O-M-E-S. Okay, I'm just going to write them all up here. Golgi, ER. Okay, so look back at this list. Nuclear envelope, lysosomes, Golgi, ER. We are going to include vacuoles. We're going to include vesicles, which are sacs that help transport things. Okay, and then you can also include the cell membrane, because the cell membrane is going to include the passage of those vesicles in and out of the cell. So inside the cytoplasm, we have this extensive endomembrane system that's going to help move things in and out of the cell. These are all related either through direct physical continuity or by the transfer of membrane segments through each of them in tiny vesicles. Okay, so for example, the rough ER and the Golgi communicate, they deal with protein production. Okay, they're not all identical, they have their own job, but they're functionally related to each other because they are all part of the nuclear envelope double phospholipid bilayer. These are true membrane-bound organelles, which is why you don't see these in a prokaryote. Okay, before we get into talking about each of these, I did want to kind of mention to you that each of these has their own little microenvironment. Okay, I'll just write it over here. If my pen, I'll just write it down here. They have their own microenvironment, which means that each organelle with a double phospholipid bilayer creates its own environment that is separate than the cytosol, which is unique because it plays a role in its functioning and what it's going to be doing. Okay, so let's start off with the ER. Endoplasmic reticulum is an interconnected network continuous with the nuclear envelope. You can see it here. Here's the nuclear envelope, and then you can see it like folds back and forth like this. This is rough. I'm just going to put RER right here. RER. And then you've got up here on this outer edges, this is SER. which is smooth endoplasmic reticulum. Both of them are composed of the same double phospholipid bilayer that comes off the nuclear envelope, but their jobs are very, very different, okay, very different. In fact, look at the rough ER. Everything that it does has to do with proteins. That's what it does, is protein production. So its job is to modify a protein. In other words, it's working with the ribosome. I forgot to mention that. The ribosome's job is to make a protein. So the ribosome is attached to the rough ER. They work together, modify and transport the protein, and it's going to send it to the Golgi apparatus. In order to do that, it's going to use these little bags that you see right here. That's a vesicle. It's a transport bubble. Okay, that's a vesicle. It's a pinching off, C-L-E, my pen will not work in that little area. It's a pinching off of the rough ER, so it's like a piece of, you can see right here, a piece of the rough ER pinches as it wraps around the protein, and it pulls off and then will travel to some other location inside the cytosol. So a vesicle is a transport sac that its job is transporting things within the cell. Okay, so that's what the rough ER does. Now I did want to point out a few things on the diagram, so I'm going to erase some of this writing right here. We're going to label a few things on the rough ER. There's a few vocabulary words. Okay, so the first one is, I wanted to mention this word called cisternae. I'm going to point to the area. It is actually the membranes and tubules. So like here, let me see if I can write on it. This little round thing right here, so like the round sac, like that, all the way around. Each little sac like this that's folded, that's called cisternae. C-I-S-T-E-R-N-A-E. That's called cisternae. That's the word for the membranous tubes and network of those folds that are on the rough endoplasmic reticulum. Okay, now you can also notice, like where my pen is, let me change colors so you can see. If I change this to black, like right in here, that looks like it's like inside space, right here, inside the cisternae. That does have another word. It's called lumen. M-E-N, okay, lumen. So it's like the environment that's inside of these sacs called the cisternae. So that's its own little micro-environment that's inside of those sacs that is very different from the cytosol. Okay, so the rough ER has cisternae, which are the folds that go back and forth, and then inside of the cisternae, we have this area known as lumen. Okay, those are a couple of vocabulary words. All right, now let's go to smooth ER. This one is, I like to call this one the multitasker. I know it sounds kind of weird, but I nicknamed him multitasker because you look up here at the rough ER and you go, oh, his job is to modify and transport a protein. That's a pretty easy job. But if you look at the smooth ER, there are a lot of jobs that this guy does. He can serve to detoxify drugs, alcohol, and poisons. That's one job. He can also take stored up glycogen, which if you remember from the biochemistry chapter, is glucose plus glucose plus glucose plus glucose. In other words, it's a polysaccharide that is stored in human liver cells and muscle cells. So it can convert that into glucose monomers. That's a pretty big job. It can, third function, synthesize, which means make lipids. Okay, make lipids. Some of the lipids that it can make are actually lipid hormones, or lipids that are needed to make our hormones, especially for guys, testosterone, or for girls, estrogen. Those are steroid hormones that we need lipids in order to make those, so one of the jobs of the smooth ER is to synthesize those. And there's actually a fourth function that's not listed down here, and it's to store calcium ions. Okay, calcium is an ion that we're going to need for proper bone growth and muscle contraction, and smooth ER's job is to store calcium ions. So look at where it is up here on the diagram. It's this, still has cisternae, and still has lumen, but not ribosomes. There are no ribosomes on that, which is what makes it smooth ER. Okay? All right. Okay, before we go to the next slide, I just wanted to mention two things really quickly on this that I wanted to hit on. The lipid part, in addition to stuff like estrogen and testosterone, let's not forget that if we need to make new phospholipids, okay, phospholipids, like if we need to replace an organelle and you would need to create a new phospholipid bilayer around that, we can make those new phospholipids using the smooth ER that we have available in the cells. And the last thing I wanted to mention is, and you're going to actually try to answer this for me, if I asked you, if you looked at these functions and you tried to name an organ in the body that would have a high concentration of smooth ER, could you think of an organ? This is an application type question. Based on these functions, what part of the body do you think would have a high concentration of smooth ER? Because each cell in your body could have more of one organelle than another. So I was hoping you'd be able to answer liver. Okay, the liver cells that we have would have a high concentration of smooth ER because one of the jobs of the liver is detoxification of drugs, alcohol, and poisons, and then also this conversion of glycogen into glucose. That's one of the jobs of the liver. Okay, go back up here. Can you name a place in the body or a type of cell that would have a lot of rough ER, which would be if you needed a lot of protein, okay, a part of the body that would need a lot of protein? There's actually quite a few places. You might have answered muscle cells, which is right, but then also there are hormones that we make. Okay, one hormone that you know really well is insulin. Let me write it over here. One hormone is insulin, and insulin is made in the pancreas, and it's a protein hormone. So what we would say is pancreatic cells would have a high concentration of rough ER because they would need to make this protein hormone called insulin. So I just wanted to hit on that a little bit. Okay, the last slide for this video, because I'm not going to make it too long, I promised you I would not make them too long. So we're going to end up with the Golgi apparatus. I want to finish with Golgi because it kind of works with the rough endoplasmic reticulum. So I wanted to end with this one. So the Golgi apparatus is sometimes nicknamed the postmaster, or I like to call it the UPS man, because the job is a lot like the UPS man for packaging and shipping. So he's going to receive the transport vesicle that comes from the rough ER. Now remember, the rough ER took a protein, modified it, and is shipping it to the Golgi in its own vesicle. So it's already coming here, it's receiving it already in a vesicle coming from the rough ER. So if you look over here at this diagram, here's the vesicle right here coming. There's one coming here, and you can see an incoming one right here. So those came from the RER, and they're coming into the Golgi. So there is actually a receiving side, okay? There's a receiving side, E-I-V-I-N-G, of the Golgi, which we call the cis space. Can you see this word right here, C-I-S? It's called the cis space. So if you look right up here, there it is. The cis space is the receiving side. The shipping side is called the trans space of the Golgi. If you just remember trans, sounds like transport. The job of Golgi is to, once it modifies this protein, it's going to ship it or transport it to another location. So the shipping aspect gets rid of it. So if you remember, the trans side down here is the side that is going to be getting rid of the final product, secreting it out in this other secretory vesicle that's going to go to its final destination. So in the case of our bodies, the rough ER would modify the protein, send it in a vesicle right here. It would come to the cis side of the Golgi. The Golgi would receive it. The Golgi would then do further modification. Okay, and then once it was done, it would send it out via another vesicle on the transphase side, which in most cases then that would go and hit the cell membrane, and then it would go out of the cell to its final destination. Now I did want to mention that some vesicles can actually go backwards from the transphase, because maybe they're not as mature, the product wasn't quite completed, and so the product comes back to the Golgi to where it can be more mature and can be a final protein that's in the proper shape. So some vesicles do leave and go to their final destination. Some vesicles come back to the Golgi until they reach maturity, and then some of them might need to go back all the way to the rough ER, where they might actually need to be used in the job, because rough ER is going to need protein to carry out its job, even though it's making a protein. So it sounds kind of weird, but it's true that this Golgi has many functions when it creates these vesicles. Some vesicles leave and go out of the cell membrane to their final destination because they're exported. Some vesicles turn around and go right back to the Golgi, and then some go all the way back to the rough ER. Okay, so does that make sense? So do you think you can say what cis side and trans space side are of that? All right, good. We're going to stop there.